All right, so we're going to talk about cell biology. Obviously, for life on Earth, as far as we know it as biologists, all living organisms are made up of cells. So a virus is not, by definition, a living organism because it is not made up of a cell. That doesn't mean that it isn't very lifelike. But as far as we understand life, it's made up of cells. Maybe on a different planet in a different solar system, life is different. But as we understand it right now, and as far as life on Earth is concerned, it's all made up of cells. And cells essentially are the building blocks of all living things. They carry out all the chemical activities needed to sustain life. So viruses cannot do these things on their own. They need to parasite a cell to be able to use the cell's machinery to replicate. They can't do it on their own, but cells can grow, divide, and do everything on their own usually. Now, there's obviously multicellular organisms that can function as tissues and organs and so forth. But again, the cells are the building blocks of all these things. It wasn't always known, cells weren't always known, for instance. They were, see if I go backwards. They weren't always known until, but they were maybe speculated. We didn't really get to know a lot until the microscope was developed. And this was done by Van Leeuwenhoek, who developed a very primitive microscope that you can see on the right, but it worked well enough to see cells. And obviously it was a delight for them to be able to collect some pond water or whatnot and look at it under his microscope and then see all sorts of little cellular organisms and so forth. This is where we start to get into understanding a little bit about cell biology. And again, it's through the aid of having a microscope. And so again, the simple microscope that you see up here above was a one lens microscope. So it's just like a glorified magnifying glass. Over here, you see which is more of a modern compound microscope. There's all sorts of different types of microscopes, dissecting microscopes, scanning electron microscopes, and so forth. But this has been the basic tool for us to understand cell biology, this compound microscope. And again, it has these different lenses and paths and objectives that allow you to see 40 times, 100 times magnification of whatever you're looking for. So this is really what pushed forward cell biology and understanding cell biology. Just to give you an idea, a lot of things that are cell related get to the point where an unaided eye can't see it. Now, that doesn't mean that you can't see a cell with your eyes. Big cells like fish eggs are really just giant cells before they've divided that you could see with the naked eye, but it's at the very bottom of our ability to see. And then that's where you start needing a light microscope. So if you see it kind of in the middle, fish eggs are kind of that bottom of our ability to see a cell. And then we need a light microscope to really begin to see the organelles, like a nucleus of a cell and all the other types of organelles. Then we get down to the nitty-gritty stuff that's really small. You need an electron microscope. And there's tunnel electron microscopes and scanning electron microscopes, and how they work is by using a beam of electrons and coating it on the outside. We actually have a scanning and, I believe, tunneling electron microscope at Western Illinois University that's allowing us to see all sorts of neat things. It was a really big deal having an electron microscope 40 or 50 years ago. And it's not used quite as much, but it's still a very, very useful tool. So that allows us to see small molecules and proteins and lipids that make up the cell membrane, as well as viruses. But we're not quite able to see an atom with them. It's not the typical tool or typical electron microscope. And then, of course, if you go to the other end of the spectrum, of course, you can see organisms and so forth. with our unaided eyes. Now, there is a limitation on cell size, and that has to do with volume. Remember that most things deal with moving through diffusion. So you're taking stuff from the outside of the cell into the inside of the cell, like nutrients and oxygen and energy containing molecules like sugars. That goes through a process called diffusion, and there's a limitation on how fast that moves. And so what happens when a cell gets too large, that diffusion rate slows down, making it harder and harder for enzyme and homeostasis and all the things that a cell needs to do to survive. Cells do much better when they stay small and have a large surface area relative to the volume of the cell. So if you look at these millimeters of cube, you can see that the bigger the box or the cube gets, the volume increases dramatically more than the surface area. And that's what this is showing you. So the surface area of a 1 millimeter cube, the volume is relatively small in relationship to the surface area. But when you get to 4 millimeter cube, the volume is dramatically more than the surface area, making again diffusion slow. So this is one of the limitations of why cells remain small. It's so it can maximize surface area to volume, and again maximize the energetics and diffusion and homeostasis and everything else that is needed. Here's the microvilli that you can find through electron microscope image. This is what would be lining your intestines on the mucosa cells. Everything's about surface area in biology when it comes to maximizing absorption of nutrients. So this is, again, maximizing your intestine's ability to absorb nutrients. Your brain is all about surface area. Everything about organisms, your lungs, is huge surface area. Remember how your lungs, if you were to stretch out the alveoli, we talk about covering a tennis court. If we're talking about, we were watching the show about reproduction, and if you were to stretch out the tubes needed to make sperm, we're talking a mile of tubes in the testes. So surface area is everything in biology, maximizes absorption. So cells have to remain small to maximize absorption of nutrients and do all the different cool things that cells do. This is to give you an idea of how sizes vary based on, so when we talk about sizes, so 20 millimeters across, we're talking your thumb tip. 2 millimeters across, we might be talking about capillaries. 0.2 micrometers, 0.2 millimeters, we're talking about tissues. 20 micrometers, micro now instead of milli, we're talking about the diameter of a cell. If we're talking 2 micrometers, we're talking about an organelle like the mitochondria. 0.2 micrometers, we're starting to talk about proteins being made with ribosomes and messenger RNA transcripts. And then we start talking about nanometers, we're actually talking 20 nanometers is about the size of a ribosome. So this is extremely small. Then not just 2 nanometers, we're able to see the shapes of atoms, and then 0.2 nanometers, we're starting to see the actual atoms themselves and not just the molecules. So this gives you an idea. And again, you really can't see much below ribosomes in most common lab equipment. So again, cell theory is the idea that all living things come from cells. Now, some virologists might point out that some cells are some viruses are very complicated, cell-like, even have a membrane. They can be relatively large and complex, and they are very lifelike, but by our definition, cells are the basic units of life, and so that is the idea. So here's the idea of cell theories. All living things, as far as we know, are made up of cells. Cells are the basic units of life, and cells only come from other cells. Now, there are some theories about how cells evolved from the first place. How did cells come to even exist? There are some theories about that. So there is a potential for cells to actually come from non-living stuff. that gets into things called chemical evolution and so forth. And the idea is that the primitive Earth had an atmosphere with some of the right components and electricity and so forth to make some primitive cells. There's other theories on how that happens as well that we're not going to get into today. But for the most part, once a cell's formed and evolved, you're not going to get this fully modern cell just de novo. It's a long evolutionary steps that before we ever had cells. So basically speaking, cells only come from other cells, or at least modern cells. They only come from other modern cells. Now, some of the ideas of cells started back in 1665 with Robert Hooke. He had looked at some, with a kind of a magnifying glass or some tool, he looked at the cork tissues of plants, and he could see little shapes to them that reminded him of honeycomb compartments called cellulae that are found in catacombs or someplace like that. So that's where the term cells come from. So they look like these little compartments to them. And so from there on out, the term cells kind of stuck, thanks to Robert Hooke's initial statement of what they kind of appeared like. Then again, as I mentioned before, Van Leeuwenhoek came up with a microscope that's basically a magnifying glass where he could look at the first microorganisms. And so we're talking 1673, not really long when you consider humans have been around for maybe around 100,000 years. We're just now beginning to understand things like cells. When human history has been much, much longer, of course. And then there's all sorts of other scientists that were involved in helping to make the cell theory. Schwann is one of the more famous ones to me because of Schwann cells that are known for coding the exon of neurons and help speed up a nerve impulse. So again, the cell theory is very important, and it started to come together around 1838 to 1839. And so this is kind of where it started off. And so remember, this is still before the Civil War. So it's in some ways a long time ago, in some ways not very long at all, when we start to get the idea of what cells are. Now, if you look at the anatomy of cells, cells are not all the same. All cells share a general structure, though. You'll see a cell membrane, you'll see a nucleus, generally, not always, cytoplasm, and a plasma membrane, which is the cell membrane. So all these different organelles have a plasma membrane. We'll get into that a little later. The cytoplasm is everything outside of the nucleus and inside the cell membrane. So that includes all the organelles as well as the watery stuff that's inside cells, known as cytosol. Now, if you look at the nucleus, there's also this part called the nucleolus that's important for making ribosomes. But generally speaking, the nucleus is a membrane-bound organelle that contains the DNA and so forth. And people might call it the brain of the cells, but remember, it's really just holding the blueprints of everything that's in your body. Again, surface area is huge. So we're talking, if you were to take all the information out of the DNA of your body, it would be equal to what you would find in a library, a very complete library when it comes to all the books and texts and everything. That's how much information can be found inside your nucleus. Now, some cells have cell walls like plants, and some cells will spit out their nucleus like your hemoglobin. It will spit out the nucleus, so it's no longer will no longer have a nucleus, but initially it would have had a nucleus. Now, the plasma membrane is basically made up of fats and lipids. These lipids are called phospholipids. So these are very famous for making the cell membrane. If you look at it, these little circles represent the phosphate side. You may not be able to see the actual chemical structure yet, but these little lines here represent fatty acid tails. So you'll see two fatty acid tails. I'll show you another picture in a moment, and a circle, and you'll see that on both sides. Well, what do you know about fats? Do they like to dissolve in water? No. So if you mix oil and water together, the oil will sit on top, right? So you have to shake it up and stuff. Well, that's what happens is basically your cell membrane is like a soap bubble. It has water on the inside and water on the outside. But the soap is also made up of fats as well, right? Soap has some fat and some parts. One side likes water and one side hates water. The outside, where those circles are right here, love water. We call them hydrophilic. The tails, the fatty parts, hate water. So they'll face each other and make this soap bubble. Essentially, cell membranes are like soap bubbles. And again, soap bubbles are made up of fats and so forth. You'll also have little proteins going through it. These proteins are like channels, they're holes that are found on the cell membrane. So if you imagine this room was a cell. That door would be a channel protein that allows stuff to come in and out of the cell. So that's what those proteins are. They're like channels that allow things to enter and exit the cell. Cell membranes or plasma membranes surround the nucleus. They surround all these different organelles. They all have this basic structure. In addition to that, on the cell surface, there are these carbohydrate structures that function as receptors and markers. So it's a way of cells to talk to each other or say, "This, I'm a human cell. This is a foreign cell." There's all sorts of things that is being communicated thanks to these little extensions that are made up of carbohydrates and proteins. So again, that's just another part of the cell surface area. Also, cholesterol, you might have heard of cholesterol, it's a fat. It is also found inside these cell membranes to help provide fluidity to the cell membrane to make it more dynamic and move around. So cholesterol is actually very important. It's not just bad. Fats aren't just bad either, because your cell membranes are made of it. If you didn't have fats, you wouldn't have cell membranes, you wouldn't have a brain, you wouldn't have cells, essentially. This is the chemical structure of phospholipids. I don't know if you remember that much about chemistry, but just realize these are atoms made up of carbon atoms and hydrogen. I hope you remember a little bit about chemistry, where these are covalent bonds, and each one of these corners would be a carbon atom. I don't know if you remember that. And it's not shown in here, but for shorthand, there'd be hydrogen atoms coming off of it. Well, this is a glycerol backbone. And again, don't worry if you don't get it all. Maybe you haven't had enough chemistry to understand this or appreciate it too much. But these are carbon atoms. You're an organic organism, meaning you're made up of carbon atoms. This is a glycerol backbone. That's what we call it. You got some oxygen atoms. O represents oxygen atoms. You have some more carbon atoms. And again, these tails are the fats. And this is the actual picture, actual diagram picture of what an atom would look like with its outer shells and electrons flowing around it. So there's different. So the other one is the chemical. So this is the chemical formula, and this is the structural formula. And then up here, you see this says phosphate. So here's the phosphate is the P. Got some oxygens and some more carbons and some nitrogens. We call this structure choline. But anyway, this side loves water, and this side, the fats, hate water. So they'll face, the water-loving sides will face towards the water, and the fatty acid tails will face internally, making a hydrophobic layer. So this is just an example of one of these here. You see that? You see the circle here? That's the phosphate and choline, and those tails are the fatty acid tails that hate water. So they're facing internally, making this hydrophobic layer. Here's another picture. So what's up here? The phosphates and the choline. The glycerol backbones here. And what are these again? Those are the fatty acid tails. So here's one layer. And then if you look down here, we see another layer, right? You see the choline and the phosphates and the glycerols. And then we see the fatty acid tails. So the fatty acid tails from both sides are facing each other. Because why? They hate water. So fats like to be with fats. And so the water will be on the outside of the cell and on the inside of the cell. So the hydrophilic sides, hydro meaning water, philic meaning love, will face towards the water. the hydrophobic tails, or the fatty acid tails, hydro meaning water, phobic meaning fear, will hate the water, so they'll face each other. And together they'll make this big sphere that makes the cell membrane, or a soap bubble, because I consider this like a cell membrane is just like a lot like a soap bubble, but with all sorts of proteins and stuff that allow for substances to enter and exit the cell. Here's another picture. Again, this would be the phosphate and the choline, and what would the red be? The fatty acid tails. So the fatty acid tails are facing each other. And then of course you have your hydrophilic heads loving water. So hydrophilic heads love water, hydrophobic tails hate water. So you form what we call a phospholipid bilayer. That is what makes the cell membrane. So we're just looking at a little segment of it right here. Here's a more complex, more complete picture of what a cell membrane would look like. Here you can see the choline and the phosphates that love the water on both sides, and the fatty acid tails in the middle. You can see some cholesterols floating around that helps make the bubble move and be fluid. You got some proteins, and some of those proteins will be like channels that allow stuff to enter and exit the cell. And you got those carbohydrate chains, and what do they do? They're a way for cells to communicate and identify themselves. So they're like signatures and messengers and all sorts of stuff. All sorts of cool stuff. Here's the proteins acting like a channel, making a little hole for things that enter and exit the cell. And then of course you got your phospholipids, and you got your fatty acid tails and your cholesterol, and then you got your phospholipid heads that love the water, so they'll face the water on the outside of the cell or on the inside of the cell. And again, the cholesterol helps the cell to move around and for the membrane to move around. Because if you ever looked at a soap bubble, it doesn't just stay in one place, right? It's kind of glistening and moving around. This picture again is also showing you the same thing. Here we have our phospholipid bilayer. You got your fatty acid tails. We just sliced off and looking at one part of it. You got your proteins, and you got all sorts of carbohydrates. So the outside of the cell is rather complex with all these different carbohydrates and messengers and ways for the cells to talk to each other. So they'll line up, and they have all sorts of important roles in communication, identification, structure for tissues to hold together. And then there's all sorts of proteins on the inside that help move things around and form the cytoskeleton that helps provide structure and movement of substances, keep all sorts of different things like that. Now, cells can also have cell walls, but not animal cells. Animals would have just the cell membrane, but plants can have the cell membrane plus the cell wall. So they got both. So they got a cell wall and a cell membrane. And then here we got inside it, the nucleus. This big thing here would be called a vacuole. It's a storage area. And again, this is the cell wall and then the cell membrane. Now, plants, because they have cell walls, need to have little holes between each of the cells. Those holes are called plasmodesma. And I think it's plasmodesmata, but it might be, just not to confuse you. The point is there's little holes between, here it is, plasmodesmata. Those are the little holes between the cells that allow for substances to move from this cell to that cell back and forth, and to share chemicals, to communicate with one another, because the cell wall would normally not allow the cells to talk to each other. And so they can still move stuff through their membranes and so forth because of these holes between these two different cells. So it's like having two rooms and you got a door going between the two. That's what the plasmodesmata would be like. Now, there are other types of cells like fungus. They will have cell walls, but they'll be made up of chitin. Bacteria will also have cell walls, but they're made up of peptoglycans. So here's what the cell membrane actually looks like. So you got the outside of the cell, the inside of the cell. This is the cell membrane. You can see there's a phospholipid bilayer. You can't really see the internal parts, but this is where the fats are inside. And then the hydrophilic heads are on the outside or the inside. The tails are facing inside. And there's all sorts of proteins and carbohydrates that are going in and out and extending beyond the cell membrane and so forth. And that's what some of these materials are out here, or structures we see out here. And then underneath it, you'll see that there is parts of the cytoskeleton. So it's kind of like a skeleton for the cell. So you can see the cytoskeleton here too. But again, it's getting to be so small. You need to have really good microscopes to see that kind of stuff. And then there's all sorts of organelles that have membranes as well. So the nucleus will have a plasma membrane too. So the plasma membrane is not only just the cell membrane, there's also the nucleus's membrane, the mitochondria's membrane, the Golgi Apparatus's membrane, the rough endoplasmic reticulum's membrane, the smooth endoplasmic reticulum's membrane, the lysosomes, and everything. All those things have a real membrane around them. And again, it'll be the fatty acid tails, the hydrophilic, and so forth. Now, again, we're going to start talking about the different organelles. Again, organelle, think about organelle. What does that mean? It's like a small organ, right? You got a cell. You have, in your body, you have organs. In the cell, you have organelles, small organ-like structures that have functions. One of them is the nucleus. So here we can see a nucleus. We cut off a part of it so you can see inside it. It would just be kind of this round structure. It has nuclear. Here's a plasma membrane with the phospholipids and all that stuff. Inside it, you would have your DNA. It's called chromatin at the time because it's all, you've seen chromosomes all coiled up. But when it's not all coiled up, it's just floating around as chromatin. So here you got your chromatin. You have a nucleolus in the center. What's the job of a nucleolus? Well, it holds on to ribosomes and so forth. It makes the ribosomes. It also helps in making, I believe, transfer RNAs as well. And then you'll notice there's little pores. Well, that allows for messenger RNA. You might not know what that means at this point, but it allows for basically messages to go out of the nucleus and tell the body how to make certain proteins. Because remember, what is the job of a nucleus? The job of a nucleus is to hold the DNA. What does DNA do? It is the blueprints of everything that your cells make, all the proteins. And for it to do so, instead of sending the whole book out to be built, because these proteins are being built inside the cell, what happens is it photocopies little stretches of genes and sends that out as messenger RNA. That would be for another class, so don't really get too bent out of shape on that. But that would lead through these nuclear pores. The nuclear pores, again, are like doors, channels made up of proteins that allow for this messages to leave. Right beside the nucleus is a thing called the rough endoplasmic reticulum. That's what the ER stands for, endoplasmic reticulum. On the next slide or two, you'll probably see the actual spelling for that. They call it rough endoplasmic reticulum because it has these dots on it. Those dots are made up of a thing called ribosomes. Ribosomes are important for making proteins. Again, it's made up of RNA. You might be confused at this point. Don't worry about that. What's the take-home message? What does rough endoplasmic reticulum make? It makes proteins. And how does it do it? It uses the blueprints that came from the nucleus. So you don't have to worry about knowing everything at this moment. Just realize those simple statements there. Don't even bother memorizing it. Just understand that. So the nucleus, the cliche would be it's the control center, the brain of the cell. But the reality is its job is to control and hold on to all this DNA, which is the blueprints of making proteins. And they'll make chromosomes. Are very important for when the cell divides and so forth. But before the chromosomes are present, like you see over here, usually it's not in a chromosome form, it's in a chromatin form, which just makes it easier for the cell to do, to make the proteins and so forth. Now, the nucleolus is this dark circle in the center. And again, its job is to make ribosomes. What is the ribosome's job? Help make protein. Again, don't worry about not understanding how that happens at this point. Just realize the nucleus is important for holding onto the DNA and then helping make ribosomes with this nucleolus. Again, there may be, I'm not sure if there's a membrane around the nucleolus, to be honest with you, but just realize there is a membrane around the nucleolus. Here's the nucleoli. Again, that is the nucleolus. Its job is to make ribosomes. And remember, what is on the rough endoplasmic reticulum that makes it rough? Ribosomes. All those little dots are ribosomes. So this nucleolus is making ribosomes that end up on the rough endoplasmic reticulum. We call it rough because we see these ribosomes. What does the rough endoplasmic reticulum do? It makes proteins. Why? Because it has ribosomes. Where does it get the DNA and the blueprints to do that? From the nucleus. So it's all closely associated with each other. Now, the cytoplasm is basically everything outside of the nucleus to the cell membrane. This includes the watery stuff. So what is the water that's inside cells called? Cytosol. So the cytosol is the fluids that are inside the cell. And then the rest of the organelles all have some type of metabolic machinery or function that's important for the cell. And we're going to get into what these different organelles do. And then there's also some non-functioning parts as well. So the first thing we're going to talk about as organelles go, and this one does not have a membrane, and it's made in the nucleolus, as I mentioned before, that is ribosomes. And there's actually two halves to it, a big half and a small half. But what is its job? It's to help make proteins. And we'll get into another class or two down the road or in another course and describe how that protein is made. But for now, just realize there's two halves of it that come together, making a ribosome, and that ribosome's job is to make proteins. What about the mitochondria of the cell? Everybody knows the cliche term. It's the powerhouse of the cell. It's such a cliche that I feel like college students should at least understand it's important for making ATP. At least add that to your vocabulary, because ATP is the fuel source of animals and plants for the most part. For most functions, ATP is involved at some level. What does ATP stand for? Well, it stands for andesine triphosphate. Andesine is a chemical. Tri means three. Phosphates are atoms. And each one of those atoms has what you call a high power covalent bond. When you pull that phosphate off, it's going to give off energy to supply towards all sorts of cellular machinery or your muscles moving and so forth. We talked about that, and we'll talk about that in animal physiology, how ATP is important for muscular contractions. Interesting enough, the mitochondria seems to have been a cell of its own sort that maybe was engulfed by a big cell in early Earth's evolution of life because it has its own DNA. Generally speaking, actually the mitochondria in your body is passed down by the mother's lineage, because when the sperm fertilizes the egg has mitochondria in it, but typically the mitochondria of the sperm stays in the tail, not the part that actually fertilizes the egg, which is going to be the head of that sperm. So again, ATP is what provides cellular energy. So mitochondria is critical for that. And it has two cell membranes as well, an inner membrane and an outer membrane. And so it does all sorts of oxidative functions to make ATP. Here's some more pictures of it. Here you can see an electron micrograph of a mitochondria. You can see its outer membrane, its inner membrane. ATP is made, and it's really neat if you take a biochemistry class, you'll get an idea of how this actually works in making ATP. So a lot of biology classes, you just get into the nitty-gritty details about how does the mitochondria really do it. Obviously, for this class, just realize mitochondria is important for making ATP. It has its own DNA, so it's almost like a bacteria-like cell living inside your other cells. Here is a sperm cell, and you'll see that the mitochondria is mostly in the mid piece of the tail, and in this actual head of the sperm, that is where the DNA is located, and that's where the nucleus is located. And so when it hits an egg, it's going to digest the outer membrane, hook up, eject the nucleus and the DNA, nuclear DNA, which is the DNA that's found in the nucleus. That's what we mean by nuclear DNA. It'll end up hooking up with the egg's DNA, and of course, then you end up with DNA from your mom and DNA from your dad. There's also chromosome. Obviously, mitochondria is really important for muscle contractions as well, or for helping with the sperm's tail to move. There's other organelles, but we won't find this in animals, but there are chloroplasts. Chloroplasts are a lot like mitochondria in their own way, in the sense that they have their own DNA, they have two membranes, but their job is to capture sunlight and convert carbohydrates, or excuse me, convert carbon dioxide into carbohydrates. So whatever carbon dioxide you're breathing out, plants will take that up through photosynthesis and make it into sugars and provide the energy needed for herbivores to live and for carnivores to eat the herbivores, and everything dies. So basically, something's eating something, but it's thanks to plants and algae and any organism that photosynthesizes, it captures that sunlight, turns that sunlight energy into food energy that the rest of the ecosystem can exist. It's thanks to these chloroplasts that life on Earth, as far as we're concerned as humans, exists. There may be some animals or organisms that have other alternative ways to making energy, but the life as we know it on Earth that we care about particularly wouldn't exist if it wasn't for photosynthesis. Here's the basic structure. We cut off a segment of the chloroplast, we look inside it. You can see there's an outer cell membrane. Again, it's going to be a phospholipid membrane. It's going to have phospholipids, just like I described. You have an inner membrane. It's going to have phospholipids. All these thycloids are going to be made up of phospholipids, and how they're all kind of structured facing each other. And then again, its job is to capture sunlight. That is another. You can learn a lot more from another class on how photosynthesis actually occurs. Here's the inner and outer membranes and the thycloids and all sorts of stuff. They think, when we talked about the evolution of cells, we had these primitive cells before we had multicellular organisms like us. We had this idea called the endosymbiotic theory. The endosymbiotic theory is the idea that a big cell engulfed some type of organelle and engulfed it and made it its own. So it could be chloroplasts, it could be mitochondria, it could be the nucleus, but basically those cells engulfed the organelles and developed this relationship, this mutualistic relationship. The big cell that engulfed the other cell, because back in early Earth's history, these cells would be eating each other, but instead of digesting it, it made it a home. So the mitochondria can live inside the big cell, or the chloroplast can live inside the big cell, divide in it, and have its own DNA. And so that's where this theory or idea came from. And so it's thanks to the endosymbiotic theory we have these organelles. Now, plants actually don't just have chloroplasts, they also have mitochondria too. There's not to confuse you, but a plant cell outside has mitochondria and chloroplasts, while animal cells only have the mitochondria. At the end, the idea is 3.5 billion years ago or so, a cell engulfed another cell, and instead of digesting it and just eating it, it started living inside it and having this relationship. Here's some cells. You can look inside it and see all the different organelles. Now let's talk about the endoplasmic reticulum. Here's how you actually spell it. Here's the endoplasmic reticulum. There's two different types. I've already told you about the rough endoplasmic reticulum. Remember what its job is and why it looks rough? Because it has mitochondria. And that mitochondria is important for making proteins. Now, what about the smooth endoplasmic reticulum? Well, the smooth endoplasmic reticulum can be found right over here. It kind of looks like funnel cakes in this picture to me. So that should remind you that its job is to break down fats, do fat metabolism. Because what we know about funnel cakes, they're deep fried, they make you fat, they're oily. So that's one way to remember that the smooth endoplasmic reticulum is smooth looking because it doesn't have any mitochondria on it. And its job is fat metabolism and detoxification of drugs. What about the rough endoplasmic reticulum? I've already said it a million times. It's important for making proteins. There's also the Golgi apparatus. Its job is to package all these proteins that the rough endoplasmic reticulum make. So here we have our rough endoplasmic reticulum. It's making those proteins from taking the DNA, making them into proteins inside it, and then they'll package it. So the rough endoplasmic reticulum will make a vesicle that looks like a soap bubble. We call it a vesicle that will leave the rough endoplasmic reticulum and travel over to the Golgi apparatus. The Golgi apparatus will modify the proteins, do all sorts of things that might need to do, and package it as a way that can be either used by the cell or leave the cell by making another package called a secretory vesicle that will float to the cell membrane and then be ejected. So this might be a way that hormones are ejected from the cell or some chemical that might be another cell might need. So we call this exocytosis. That would be something for another class as well. And basically this soap bubble is made up of plasma membrane. So have you ever seen two bubbles come together? And when they come together, it becomes a bigger bubble. Well, imagine a little bubble joins the big bubble. Well, that opens up and whatever was in that soap bubble gets ejected, and that's called exocytosis. That's again, probably for another class. Here's another picture of the rough endoplasmic reticulum. Notice those dots. That is mitochondria. What is its job to do? Everybody should know. It makes proteins. What does the smooth endoplasmic reticulum do? Metabolizes fats. Here's a real picture of it. This is an electron microscope picture. You can see the mitochondria in them. And then you can over here, you can see the smooth endoplasmic reticulum. And then what does the Golgi apparatus do? Modifies the fats, proteins, or whatever is being made. And then it can be secreted or modified inside the cell or do all sorts of stuff. Here it is being secreted. It leaves as a soap bubble, even though we call it a vesicle, secretory vesicle. And it exits by attaching to the cell membrane and becoming part of the cell membrane. So this vesicle becomes part of the cell membrane. Here's a real picture of the Golgi apparatus. And here's real droplets coming in, joining, being modified, and leaving. So here you can see the Golgi apparatus. You got, well, it says secretory vesicles here. We'll have vesicles coming from the endoplasmic reticulum, attaching. Then they'll be modified inside the Golgi apparatus and then secreted and go to that cell membrane. Here's the actual picture of it right here. And then you can see the different vesicles. Again, those are phospholipid membranes. So you can see it coming out, being secreted. And we call that exocytosis. Here's another picture of it. Here you have your chromatin, because the chromosomes would be inside here, but it's all laid out flat, so you can't see the chromosomes except during cell division. You got your nuclear membrane, you got your rough endoplasmic reticulum. So messenger RNA or the transcripts or the photocopy of the DNA will enter into the rough endoplasmic reticulum, hook up with the ribosomes, get made into proteins. And then from there, the proteins will go through it with a transport vesicle from the rough endoplasmic reticulum to the Golgi apparatus. Again, the Golgi complex or apparatus's job is to modify those proteins and then through secretory vesicles, it'll travel to the cell membrane and join. Because remember, this is a plasma membrane. We slice through the top. It will join with the cell membrane and be secreted outward. Or it might hook up with another vesicle to be modified even further, or a lysosome, which might break it down. So there's all sorts of different potential inroads for these proteins that were made. The lysosomes are another type of vesicle that contains enzymes that will digest non-usable materials from the cell. So they will help to break down proteins or whatever needs to be broken down. Peroxisomes are membranous sacs that are used to detoxify harmful substances and break down harmful chemicals or free radicals. Remember, free radicals are basically a chemical that can run around grabbing electrons and really causing havoc on cell membranes or biological proteins and stuff like that. So they're very harmful, free radicals. So we try to break down free radicals by making them less harmful. And so that's what peroxisomes' jobs are to do. How do cells divide? They replicate by pinching in half. And in future classes, we'll talk about how that happens. So again, peroxisomes can degrade hydrogen peroxide by using an enzyme called catalase, which will make it less harmful, make the free radicals less harmful. Plants have them, and they can convert fats to carbohydrates for seed germination. So peroxisomes may have different roles, whether we're talking about plants versus animals. Photorespiration causes the potential for free radicals, and so they'll help in detoxification. While animals, when they're eating, will run across lots of harmful chemicals, detoxifying ethanol, for instance. Peroxisomes are responsible for doing all those kind of things. They're found in the livers and kidneys in higher amounts where a lot of detoxification takes place. You'll also see proteins running around cells, and their job is to function as a cytoskeleton. So they'll help hold organelles in place and move and shuttle proteins and vesicles around. So they have multiple functions inside cells. They help keep a structure and help move particles around. They're also very important when we're dividing the cell into two. So here's pictures of cytoskeleton. So if we removed out the outer side of the cell, you'll see the cytoskeleton surrounding all sorts of structures. You can even see the shadows of where the nucleus would be. So again, cytoskeleton helps to keep the structure of your cell, but also to shuttle, kind of like a conveyor belt or kind of like an escalator or whatever. It will help move substances around the cell, function like a moving sidewalk. So if you go back, I'm going the wrong direction maybe. Go back the other way. So here's the cytoskeletons. You can see the different microfilaments. They're made up of just proteins. Often these proteins are called actin. So each one of these circles are a actin, and they are the most easily among the most prevalent proteins that you'll find in the cell of any cell, and have all sorts of jobs, from even important for movement of muscles, these actins. And so the actins will circles that will glue together. And when I say glue, I mean covalent bonds and all sorts of things. There's microfilaments, microtubules. Again, a lot of them are made up of these actins. And then here you can see the cytoskeleton holding the different organelles in place and so forth. Here's more of those subunits and microfilaments and tubulins. There's more of the microtubules. The centrioles are important for moving the microfilaments around and help in, particularly during mitosis. Mitosis is cell division, and that's again for more of a future class. Again, all organisms that are living are made up of cells that we are aware of. In fact, we even classify organisms based on the type of cells they have. Eukaryotes means true cells are us. So we're made up of all these things like animals, fungi, plants, and protists are all eukaryotes. And so they're under the group Eukarya, and you can see it highlighted in blue. Then there are these bacteria that live in extreme environments, hot sulfur springs or very acidic environments, all sorts of really crazy, dangerous environments. Maybe even can survive living on an asteroid, all sorts of things crazy like that. Those are known as archaea, and they have a cell that's quite a bit different, uniquely different enough that it gets its own domain. Now, just to make sure you understand what a domain is, remember there's kingdom, phylum, class, order, family, genus, species. So animals is a kingdom, domain is even a greater, bigger group. Then you have your common bacteria. Those are your eubacteria. These are things like E. coli, most of the things that you'll learn in your regular microbiology class. And again, their cell properties, the type, and we'll get maybe another diagram that describe it, put them in a different domain. So there's enough differences between archaea and bacteria, though we might think of them as both bacteria, to put them in different domains. So that means there's a huge, huge evolutionary change in time, incredibly long distance. And then eukarya, which is actually more similar to archaea, evolutionarily, even though we're still talking billions of years ago, have a true nucleus. Bacteria doesn't have a nucleus. Archaea does not have a nucleus. Bacteria doesn't have organelles like mitochondria. Archaea don't, but true cells do. And so this is just kind of showing you the different properties of these different cells. Like I said, organelles are present in eukarya, a nucleus, microfilaments, all that stuff, actin, exocytosis, endocytosis, mitosis, and meiosis. All that happens in eukaryotic cells, but doesn't happen in prokaryotic cells, whether they be eubacteria or archaea. The RNA is different. There's lots of things that are different about it. And the most obvious and most common thing to see is there's no nucleus inside bacteria. Here's the structures of cells. You can see there's no nucleus. There's a cell membrane. They often have cell walls, but you don't see the other stuff. Here's different bacteria again, spirally. And then let's talk about plants and animals. Plants and animals are both eukaryotic cells. They both have a nucleus. They both have rough endoplasmic reticulum, smooth endoplasmic reticulum. The plants have big vacuoles. Animal cells don't. Plants have chloroplasts. Animal cells don't. They both have mitochondria. Plants have big cell walls and cell membranes. Animal cells don't have cell walls. There's plasmodesmata between the cell walls. Obviously, animal cells aren't going to have that. And again, the vacuoles are usually a big storage toxic area, depending on what it's used for, that are found in plant cells. We don't really see those big vacuoles in animal cells. So again, animal cells are more like this. This is kind of the classic animal cell, generalized circular shape. But the reality is animal cells come in lots of different diversity. And so here's all the organelles that we described. Vacuoles are a lot smaller. But the cell diversity of animal cells is great. There's a lipid droplet in a fat cell. So its whole job is to hold on to lipids. And then here's macrophages. These are cells that run around eating other cells. So they're part of your immune system, or as you might call them, the white blood cells. They have pseudopods, so they're like amoebas that'll run around and eat other cells that aren't supposed to be in your body. And they use these lysozymes to dissolve those other cells. The erythrocytes don't have hemoglobin. There's no organelles in erythrocytes. Mature ones, I should point that out. Young ones will have a hemoglobin, will have a nucleus and make lots of the hemoglobin first, and then that nucleus will be destroyed or expelled from the red blood cells. There's also cells that are important for making the fibers and the bones and the lay down the skeleton. There's epithelial cells as well that line up your organs and body coverings. Here's a red blood cell and it's concave shape, so it's kind of like a lifesaver or a donut almost. It's not hollow, but it's got a big outer tube, kind of like an inner tube, an inner, and again, the nucleus has been kicked out. We also have large, long cells. They're not really so much large, but they're very long, and they'll join together to make nerve bundles. And so this is what an example of a nerve cell fiber would look like, with a long, long axon and these axon terminals and dendrites in a cell body. But these would extend from your back down your leg. So these are really long cells, even though, again, they would be hard to see. Here you got your sperm cell and your flagellum, and you have your egg cell over here. So sperm cells are a lot smaller than the ovum, even though it's not shown as being that different in this picture. Here's the start of a nerve cell. Here you got your nucleus. You can kind of see where the nucleus would be. I think it's mostly showing you the cytoskeleton, and then the axon would travel from this picture way down to the floor. So you're seeing just the cell body mostly, and then these were the dendrites and so forth. Here's the muscles cells. These are the contractile units and the nucleuses and so forth. Here's smooth muscles with the different spindle fibers and so forth. And then we have our cytosol and our cell walls of plant cells. So this is your typical plant cell, but even they come in all sorts of different structures and sizes. There's going to be a lot of diversity in those as well. Big vacuoles, again, temporary. They can be used for long-term storage in plants, but they're only small and temporary in us. Protists might have them too. Here's your Plasmodesma. Again, they're just the pores between cell walls, so that particles can go from one cell to the next. They're like doors between cells. Here's some more pictures. Look how thick the cell wall is of the plant cell. And then, of course, there's the inner membrane, but there's a secondary wall and a primary wall. And then you're going to have that inner cell membrane. Well, anyway, I hope that gives you an idea about cells. Here's muscle cells. I'm not sure. Lots of contractile units made up of those actin proteins. So anyway, I'm hoping you have an idea about cells that maybe either refreshed what you should have learned in high school, or helps you to just remind you because it's been a long summer break. But these are important things that every biologist should at least be familiar with, regardless if they become a wildlife biologist or work in a zoo. Any educated person should have some basic idea of what I just described today. So I hope you'll take a little time to learn it. Read through the notes, watch the video again, watch a lot of other videos, because there's lots of great stuff on YouTube to help reinforce this. Anyway, thanks for listening, and good luck with your classes. Take care. Bye.